Hello everyone, this is Harshavat. Welcome to Visionary First Step YouTube channel, and today we are going to discuss about one of the important topic, that is biological classification. As we all know, first unit is all about classification. Living world deals with the nomenclature of the organisms. Biological classification totally about the foundation, how the classification system will work, and plant kingdom and animal kingdom are the extension of this topic, a part of this topic, okay, separately given. Now we will try to understand what is discussed in the topic biological classification in more detailed way, which will help you to understand for theory exam as well as for the competitive exam. See, this topic includes kingdom Monera, nothing but the bacteria, kingdom Protista, all the unicellular eukaryotes, fungi. We all know fungi mainly includes mushroom like organisms or bread molds, which are commonly observed. Then Plantae, plants, Animalia, animals. Okay, so this 5 kingdom system was proposed by R.H. Whittaker. We will study in detail in the further topics. Here, viruses, viroids and lichens and some other prions are not included in 5 kingdom system of classification, and that we will study at the last part of this topic. Okay, now just enjoy this slide. This is simply to show how the diverse organisms are present. Look at this single-celled organism, that is bacteria, and this is also a single-celled organism, and this is paramecium, here fungi, mushroom, and this is bougainvillea plant. You all know it produces very beautiful color. Now this here, because it is producing many type of flowers because of grafting, and this is the lion. Just enjoy this slide. Now coming to the topic, initially many attempts were taken to classify the organisms. Okay, so how this classification idea got started, actually it is not scientific initially, because of the need, daily usage, like we need organisms like plants and animals as a source of food. So the classification got started on the concept of having food, shelter and clothing. Aristotle, he was first to try to explain the basic concept of classification, where he just classified the plants like herbs, shrubs and trees. Now tell me, which organism we consider as a herb? The plant which have ability to grow up to 2 to 3 feet height and their stem is very weak, they are considered as herbs. Shrubs, they may go up to 5 to 6 feet height and they are profusely branched and they are considered as shrubs. And you know, having a woody stem, then we can call them as a trees. So that is the basic classification of plants by Aristotle. Then he classified animals as animals with red blood and animals without red blood. Now, have you seen any organisms without red blood? Yes, we usually observe this. Whenever a cockroach in our bedroom or in the house, when we hit them with the strong material, they just burst open and produces a white color fluid, right? So those are the organisms without red blood. Okay, so insects usually they don't have red blood, and other organisms, most of them, they have red blood. So based on the presence and absence of red blood, he classified the animals. So that is the basic in which Aristotle classified the organisms. Now, this was just a not completely scientific, it is just a way of classification. Then Carolus Linnaeus approached this classification system in a more scientific way. So what he did here, he classified the plants and animals as Plantae and Animalia under two kingdom system of classification. Okay, so here this system of classification was very primitive, so very few characters are considered for the classification. Now, the organisms with the cell wall, without knowing its composition, suppose organism contains cell wall, they are all classified under plant. Now imagine bacteria also has cell wall, then fungus also has cell wall, and some of the Protista members also has cell wall. They are all placed under Plantae. Okay, and animals without cell wall are placed, organisms without cell wall are placed under Animalia. So that was the crude idea towards, but scientific approach towards the classification system by Carolus Linnaeus. Okay, so this system has some limitations. Those limitations are given here. Now, no difference between prokaryotes and eukaryotes. Now we will try to understand what is the basic difference between the prokaryote and a eukaryote. You know, prokaryon represents primitive nucleus. Karyon means nucleus. Primitive nucleus means nucleus is not surrounded by nuclear membrane. That means genetic material is directly present in the cytoplasm. Eukaryotes means they have true nucleus, membrane bound nucleus. They are considered as eukaryotes. Now just imagine here bacteria and other organisms are placed together in Plantae. How we can differentiate prokaryote and eukaryote? That was one drawback. Second, no difference between unicellular and multicellular organisms. So this is one more thing. See, bacteria, unicellular organisms. Then diatoms, nothing but Protista members, chlorella, chlamydomonas, then paramecium, amoeba, all these are unicellular organisms. Now there is no boundary, he directly placed under Plantae. Okay, and no difference between photosynthetic and non-photosynthetic organisms. He has to consider all these criteria to have a better classification, but these were not considered, so these are the drawbacks. Photosynthetic organisms can prepare their own food, they are autotrophs actually. Non-photosynthetic organisms are heterotrophs, so this is very important character in classification of the organism, but that were not considered here. Now here what he need to consider at the time of classification actually, that is cell type. Okay, then cell wall, cell type in the sense prokaryote or eukaryote, cell wall, whether it is made up of cellulose or it is made up of chitin or it is any of the non-cellulosic material, that he should consider, like peptidoglycan and murin. Then nutrition mode, just now I said autotrophs, heterotrophs. In autotrophs again photoautotrophs, chemoautotrophs. In heterotrophs, parasites, pathogens. Like there are so many mode of nutrition is there that has to be considered at the time of classification. Now habitat, where they live, whether they are freshwater forms, marine water forms, terrestrial forms. So this they have to consider. Then mode of reproduction, vegetative mode of reproduction, asexual mode of reproduction, sexual mode of reproduction. If there is in sexual mode of reproduction, again there are so many categories are there, that has to be considered. Then evolutionary relationship, that is what we call it as phylogeny, evolutionary relationship. So one organism is highly evolved means we should trace their ancestors. That will give one strong basis for the classification. Okay. Now, if we just observe the different classification here, over a period of time, concept of classification gradually got changed. Carolus Linnaeus proposed a two kingdom system of classification, but there is no proper classification of prokaryotic organisms and eukaryotic organisms, unicellular organisms and multicellular organisms, photosynthetic organisms and non-photosynthetic organisms. But later scientists, knowing the importance of this, they considered those characters and proposed three kingdom system of classification by Ernst Haeckel. So along with the Plantae and Animalia, he included Protista. So what was the criteria taken by Ernst Haeckel to propose the kingdom Protista? All unicellular eukaryotes. Very, very important character. Unicellular eukaryotes. Okay, so there is a clear boundary: unicellular prokaryotes, unicellular eukaryotes. Okay, then just now I said, then prokaryotes need to be placed in some of the classification. By knowing that importance, four kingdom classification were proposed by Copeland. He included Monera, so that includes the unicellular prokaryotes. Of course, all the prokaryote members are unicellular only, no doubt in that. Okay, so this is how now we have four kingdom system of classification, which includes Monera, Protista, and then Plantae and Animalia. Now there was one problem here. Fungus having chitinous cell wall are heterotrophs, totally heterotrophs. They are different organisms. Now there is no specific classification to place the mycota or fungi. By knowing this importance, R.H. Whittaker proposed the five kingdom system of classification. That included Monera, Protista, Mycota, Plantae and Animalia. Just observe the pattern of classification here from the two kingdom till the five kingdom system of classification. Animalia and Plantae remained as it is, and they included more and more kingdoms like Monera, Protista and Mycota. Okay, so that was the basis. Now this chart clearly explains the characters taken for the classification of the organisms. Very important table. We try to understand each and every word. So this is the R.H. Whittaker's system of classification. Now when it comes to cell type, here just observe Monera, they are prokaryotes. Except Monera, all of them are eukaryotic. They have true nucleus. There is a clear difference between Monera and other organisms. So these are the primitive organisms. Now coming to body organization, of course all Moneran members are unicellular. There is no exception. Coming to Protista, mostly, not mostly, okay, they are unicellular, because the criteria itself is unicellular eukaryotes. Okay, they are all unicellular. Okay, then fungi are multicellular, and here we observe a loose arrangement of tissue. Okay, so tissue can be observed in fruiting bodies. Now in Plantae we observe tissue and organ, parenchyma, collenchyma, sclerenchyma. These are the tissues. When it comes to organ, xylem and phloem act as an organ of conduction of water and food. Now in the case of Animalia, we observe tissue, organ and organ system. Of course, we know we have organ system, well coordinated organ system. Without one we cannot survive. Without brain we cannot survive. Without heart we cannot survive. Without kidney we cannot survive. So all are interlinked. That is what we call it as organ system. But in primitive Animalia we observe tissue level, and then we observe organ level. most advanced type of animals, they show organ system level of organization. So it is a broad classification and the criteria for that. Now coming to cell wall, very important characters. This we need to remember. In the Monera, they have polysaccharide plus amino acid. You know what is polysaccharide? It is a chain of sugars. Polysaccharide plus amino acid, that forms the peptidoglycan or murin. Please remember the name, peptidoglycan or murin, and that is present here in the case of Monera. In Protista, you cannot define the exact cell wall. The reason is, now the organism is taken from fungus like Saccharomyces cerevisiae, that is yeast. Organism is taken from Plantae like Chlamydomonas and Chlorella. They are exactly like plants but taken for the criteria that the unicellular eukaryotes to the Protista. Now some of the animal like organism like amoeba, entamoeba and paramecium, they are all taken for the kingdom Protista. Now here, how can you say the exact cell wall? This is the cell wall. There is no boundary like that. So here given present in some of the organisms. Like Chlamydomonas has cellulose cell wall. Okay. Yeast has chitinous cell wall. So that is how in some we observe the cell wall. Now in fungi, the criteria for cell wall is chitin, very important. Okay, chitin is the cell wall material. Then in Plantae, cellulose. In animals it is absent. Animals don't have cell wall. Now mode of nutrition, all the technical terms are very important, we need to remember. Okay, we'll try to understand all the words one by one. Autotrophic, meaning they have the ability to fix the atmospheric sunlight and then they can convert them into a form of energy. They are autotrophic. Chemoautotrophic and photoautotrophic. Chemosynthetic, photosynthetic. See, there are some bacteria like Nitrosomonas, they have ability to fix the chemical substance and from that they derive the energy. Okay. Autotrophic, anyhow we know Nostoc and Anabaena are the autotrophic bacteria. They have a pigments through which they absorb sunlight and synthesize their own energy. Then heterotrophic, most of the bacteria are heterotrophic again. Though some of them are parasites, some of them are pathogens. They cause disease like cholera, tetanus, typhoid, in plant citrus canker. All these are caused by bacterial disease. Okay. Then they are saprophytic, means growing on dead and decaying material. Okay. So very important character. In Monera we observe most diverse form of mode of nutrition. Okay, that is the very important character. Now coming to Protista, so some of them are autotrophic, some of them are heterotrophic. Just now I said, because this classification doesn't have a proper boundary. So here we observe autotrophs as well as heterotrophs. Now coming to fungi, they are exclusively heterotrophs. There is no exception where they show an autotrophic mode of nutrition. All of them are heterotrophs. But in heterotrophs, there are many types again. Some of them are saprophytes, growing on dead and decaying material. Some of them are pathogens. They cause disease. Okay, ringworm caused in human beings are by the fungus. Okay, it is a pathogen. And some of them are coprophilous, growing on the dung of the animal. So there are different types in heterotrophic mode of nutrition. And coming to Plantae, exclusively they are autotrophic. They can synthesize their own food. Some exceptions are there, like Cuscuta. It is a total stem parasite. There are some organisms, they are partial parasites. Other than that, all the plants are autotrophic. Animals, here again there are some important character. They are heterotrophic. They show holozoic mode of nutrition. Very important. Holozoic means ingestion. That means digested food are taken to the cell. That is ingestion. That we call it as holozoic mode of nutrition. Now coming to nuclear membrane, this is again a very, very important character to form a clear difference between prokaryote and eukaryote. Okay, nuclear membrane is absent in Monera, and all the classifications nuclear membrane is present. Now coming to examples, for Monera, example is bacteria, Protista, paramecium, amoeba, entamoeba, and dinoflagellate, desmids, diatoms, all these are the examples. Then coming to fungi, mushrooms, trichoderma, then rhizopus, these are the examples. And Plantae, you all know, algae, bryophytes, pteridophytes, gymnosperm, angiosperms, they are all plants. And in Animalia, chordates and non-chordates we will consider. Okay, so that is about the R.H. Whittaker 5 kingdom system of classification. Now, so same thing is given here. R.H. Whittaker in 1969 proposed 5 kingdom system of classification, and kingdoms defined by him were named Monera, Protista, Mycota, and Plantae and Animalia. The main criteria for the classification already we discussed actually: cell structure, body organization, mode of nutrition, reproduction, phylogenetic relationship. Okay. Now, this is very, very important. Till now, actually, we were discussing about 5 kingdom system of classification. But here, one more classification system is there, that is called 3 domain system of classification. What is this 3 domain? Here, bacteria, they classified them as archaea and eukarya. There is a reason for that. This archaebacteria are the group of bacteria, they have ability to survive in most extreme conditions. So they are separated because their cell wall is different from the eubacteria. And eubacteria are the very common bacteria, they grow in normal habitats. Okay, so this is how bacteria itself, Monera itself, classified into 2. And all the eukaryotes are classified separately. So archaea, eukarya, and archaea, bacteria, and eukaryotes, or you can say eukarya. This is how 3 domain system of classifications were introduced. So if we consider 3 domain for 5 kingdom system of classification, then it will become 6 kingdom system of classification. Okay, but this was not in practice. Now, issues and considerations that influenced the classification system. This already we discussed. What is the reason for 3 kingdom system of classification, 4 kingdom system of classification, and 5 kingdom system of classification. So we'll go in brief here. See, when they got to know that prokaryotes were not placed according to their characters, they proposed kingdom Monera. Okay, so that is the 3 kingdom system of classification. Once Monera kingdom was formed, again when they start to realize that unicellular eukaryotes were placed randomly, so by knowing the importance of that, all the unicellular eukaryotes were placed under Protista. That is how 4 kingdom system of classification was developed by Copeland. And then again, while researching, R.H. Whittaker get to know that fungus were not given a proper classification. So he introduced Mycota. That is how 5 kingdom system of classification developed, eventually by knowing the importance of placing the particular organisms in a particular group. Let us continue with the topic, kingdom Monera. You know, Monera is all about bacteria. Can we see bacteria under our naked eye? No, it is not possible. But we can observe the disease caused by bacteria and its symptoms. Bacteria causes disease in animals, bacteria causes disease in plants. And sometimes we observe that if you keep the food for certain interval of time, colony of bacteria grow on the surface of the food, and that spoils the food. And we know that every day we consume millions of bacteria through curd or buttermilk. Okay, so that is a source of bacteria for our body. Remember, from mouth to anus, we require all the type of healthy bacteria which help us to digest the food. Because of bacteria we are living, that is more important. Okay, they are helping us to maintain our health even on the surface as well as inside the body. Now, coming to the topic, just observe this picture. There are so many details. The structure of bacteria we study in detail in Cell the Unit of Life, but here I will go through certain parts. Let us try to understand the structure of bacteria first, and then we go to the characteristics. Now, bacteria consisting of cell wall. Now, cell wall is made up of peptidoglycan, or we can say murin. This peptidoglycan is made up of polysaccharide plus amino acid. Now, next to this cell wall, it has plasma membrane. Here you can see the different layers. The outermost is the capsule. Now, there are actually different thickness of the capsule depending on the virulence nature of the bacterium. Capsule provides actually protection to the bacterium because it acts as a pathogen. It has to survive in an environment. It is very harsh. So to protect its cell, it has a capsule. Okay. So based on that, we again classify them as gram positive and gram negative bacteria. That again we'll study in Cell the Unit of Topic. Then next to cell capsule we have cell wall, and that is made up of peptidoglycan. Then plasma membrane. Plasma membrane is similar to eukaryotic organisms but shows some foldings, and that we call it as mesosomes. It performs different functions like respiration, excretion, etc. Okay. Now here on the body of the bacteria you observe many hair-like structures. The small hair-like structure are called pili, and slightly larger one is called fimbriae. Actually, this fimbriae helps the bacterium to adhere to the substratum. Okay, substratum in the sense it may be rock, it may be soil, it may be body of the organism, whatever it is, that is the substratum. So fimbriae helps the bacterium to attach to the substratum. Now, what is the function of pilus or pili? Very important. See this pili, this one helps during conjugation. Now, what is conjugation? Bacteria usually reproduce by binary fission method, but they also sometimes show sexual mode of reproduction. At that time, two compatible bacteria, they come close to each other and they form a conjugation bridge with the help of pilus or pili, and they exchange the extra chromosomal genetic material, that is plasmid. Okay, this is very interesting mode of reproduction observed in bacteria. And flagellum helps in locomotion. The number of flagella will vary. It is not same in all the bacteria. Some bacteria has single flagella, some have two, and some have many flagella over the body. So that is how flagellum will vary. Now inside the plasma membrane, it has cytoplasm. There is no membrane bound organelles here. There is no mitochondria, there is no endoplasmic reticulum. Those structures are not observed here. But it has ribosomes, and they are of 70S type. Okay, and nucleus is present freely in the cytoplasm, so it is called nucleoid. Okay, this is the linear DNA. Along with that, it also has extra chromosomal double-stranded self-replicating DNA, and that is called plasmid. Plasmid gives some antibiotic resistance property to the bacterium. Okay, now so this is about the structure of bacterium. Now coming to general characteristics, it's very important. Where do we commonly observe bacteria? Just now I said we cannot see through the naked eye, but we have to observe those material under microscope. Now, if you take a handful of soil, you get to know different types of thousands of bacteria. You take any sample of food, there is a bacteria. Any part of the surface of the body, there is a bacteria. Now, why these bacteria are found everywhere? They are cosmopolitan in distribution and they have a ability to survive in different habitats. Okay, I just told they have diverse mode of nutrition. Okay, now they are the most abundant microorganisms. They found almost everywhere and live in extreme habitats. Extreme habitat, have you seen any extreme habitats? Now just imagine keeping your body in the ocean for 1 hour or 2 hours. What will happen? Your body starts to show the exosmosis and cells starts to shrink by the loss of water. But bacteria can survive in such environment in extreme salty conditions. We can say lagoons. Lagoons are extreme salty conditions. Okay. At the same time, hot water spring where the temperature of the water is very high may reach up to 100 degrees Celsius. In such environment, we can see bacteria. And in the snow, very cold environment, there also you can see the bacteria. So in extreme conditions also we observe the bacteria. Okay. Some of them are parasites. We'll discuss more about that in the detailed classification of bacteria later. Now coming to the classification of bacteria based on its shape. Here just observe the round shaped coccus, or you can say cocci. Okay. And these are the rod shaped bacterium and we call it as bacillus. And this is spiral shaped spirillum. And here you can see comma shaped bacteria and they are called Vibrium or Vibrio. So these are the classification of bacteria based on its shape. Usually in competitive exams, these type of concepts are taken for match the following type of questions. Okay. Now coming to characters. Just now I said they can survive in extreme temperature, extreme salt habitat. Okay. And some of them are phototropes, some of them are chemoautotropes, some of them are heterotropes, some of them are parasites, some of them are pathogens. You observe a diverse mode of nutrition in bacteria. Some of them are saprophytes also. Now coming to the classification, bacteria are classified as archibacteria and eubacteria. Now what are these archibacteria and how they are different from eubacteria? Archibacteria are the group of the bacteria. They have adaptation to survive in extreme environmental conditions. Why? There should be some difference compared to eubacteria. Then only they can survive in the extreme temperatures or extreme conditions. That is the difference in their cell wall composition. So compared to eubacteria, the cell wall composition of the archibacteria is slightly different. Okay, so that is how they can adapt it to survive in extreme conditions. So here they are classified into 3 groups. One is thermoacidophils. What are these thermoacidophils? Thermo means something related to temperature. Now, if they are adapted to survive in extreme high temperature and extreme low temperature, hot spring has extreme high temperature, snow has extreme low temperature, and if they adapted to survive in these extreme conditions, then they are called thermoacidophils. One more term is added in that, acidophils. In the sense, consider your stomach, where the environment is very harsh. It is acidic environment. HCl is present in your stomach, but still bacteria will survive. Like Helicobacter pylori, they survive in our stomach in that extreme acidic conditions. So all these bacteria having ability to survive in extreme high temperature, low temperature and extreme acidic condition are classified under thermoacidophils. Okay. Then some of the bacteria are classified as methanogens. See, this is very interesting. If you go to any village part of the country where they grow cattle, what they do, they collect the dung of the cattle and they produce the biogas. We know that biogas is nothing but the methane gas. How this methane gas is produced? It is because of the bacteria methanogens. Remember, ruminant organisms. Ruminant organism means the organisms which are having four-chambered stomach. We know while cattle's grazing, they take food very fast, and after that they create the bolus, and once again they chew it and they digest it. So this is how the ruminant organisms they digest. These ruminant organisms in their gut, they have this methanogens. Okay, methanogens, they help in formation of methane gas. Okay, and one more type is there, that is halophils. So these are the bacteria have the ability to survive in extreme saline waters. Just now I said lagoons. You know what is lagoon? See, there is one sea, and the shore of the sea is having very large rock, consider that, and when the tide comes and hits the rock, this water will be moved to the next part of the rock, and there it forms a small area where water get accumulated for many years, and the water evaporates and starts to concentrate. It is highly salty. So that is the lagoon. In that area also you can observe the bacteria. They are called halophils. Okay. So all these organisms are surviving in extreme habitats. Now coming to eubacteria. So these are some common bacteria. Very common bacteria example for eubacteria is photoautotropic bacteria like nostoc and Animalia. Here the picture is given, just observe. There are some details about this picture. Here you can see the organisms. These are unicellular. Actually this unicellular bacteria are forming a colony. So this is the bacterial colony. Bacterial colony. Okay. They are photoautotrophs. By using the sunlight, they can synthesize their own food, but they are together. Here you can see some of the specialized cells, that is called heterocyst. This heterocyst have the ability to fix the atmospheric nitrogen. That means whenever sunlight is deprived, sunlight is less or there is no sunlight, at that time this colony requires a food or source of energy. So some of the cells of this colony having ability to fix the atmospheric nitrogen, at the time of unfavorable condition when sunlight is not there, they help in fixing the nitrogen and deriving the source of energy. So that is called heterocyst. Now if you look at this colony, it has mucilaginous sheath. Okay, it has a mucilaginous sheath. Do you know what is the role of mucilaginous sheath? These bacterial colony are directly exposed to sunlight. When they are directly exposed to sunlight, there is a high chance the colony get desiccation or completely dried up. It may kill the bacteria. So they have a mucilaginous sheath which will protect the colony. Okay, so that is about the nostoc. So nostoc and Animalia are the example of autotropic bacteria, eubacteria. Now coming to other examples. Now here remember they have a chlorophyll which is similar to green plants. Now chemosynthetic bacteria, that means bacteria which have the ability to derive the energy by using the chemicals, like nitrogen. Nitrosomonas is a type of bacteria which convert the nitrogen to nitrite and nitrates and derive the source of energy. Similarly, we know that organisms helps in recycling the chemicals present in the decomposing cells, like phosphorus is recycled, iron is recycled, nitrogen is recycled. It is because of the bacterial activity. Now those bacteria uses the chemicals as a source of energy to derive their food. Okay. Then there are some bacteria, they are of course heterotrophs. Now I take one example. We study this in next topic also, in morphology topic. In leguminous plants, in their roots, they form a nodule, pink color nodule. This pink color nodule consisting of rhizobium bacteria. Rhizobium bacteria is a heterotroph, cannot synthesize their own food, but it takes the shelter in the roots of the leguminous plant, because roots of the leguminous plant is favorable condition for growth of the rhizobium, where it produces a leg hemoglobin, which is the oxygen scavenger. Inside the nodule, it creates the anaerobic condition which favors the rhizobium bacteria to use its enzyme to fix the atmospheric nitrogen. And here, why it fixes atmospheric nitrogen? Because it derives the energy from it, not the source of food. It gets the source of food from the leguminous plant. The byproduct formed like nitrites and nitrates at the time of nitrogen fixation, that is used by the plants, because plants require nitrogen for their normal growth. Okay. Now there are some of the heterotrophic bacteria are pathogens, they cause disease. Not only parasites, some of them are pathogens. Now we know that cholera, tetanus, typhoid, citrus canker, these are caused by the diseases caused by the bacteria. Okay, citrus canker is caused in plants like lemon tree. We observe the citrus canker disease. The small lesions are formed on the fruits, okay, and the leaves. Now bacteria, they reproduce generally by binary fission. That is the most common mode of reproduction observed in bacteria. If not, then they reproduce by the sexual mode of reproduction, where already we studied pili, it forms the conjugation bridge through which extra chromosomal circular double-stranded self-replicating DNA get exchanged and mode of sexual reproduction takes place. Okay, that is the DNA transfer from one bacterium to the other. The last and very important part of the topic, kingdom Monera, that is mycoplasma. See, mycoplasma, even though it belongs to kingdom Monera, that is bacterium, these are special type of organisms. They are the smallest living organisms known. Okay, so what are the important characters? They lack cell wall. Actually, bacteria, they have cell wall, and composition of cell wall slightly changes, but majorly it is peptidoglycan or murin. But here in mycoplasma, they lack the cell wall, and they are the smallest living organisms known and survive without oxygen. That means it is not obligatory. Sometimes without oxygen also it can survive. And many mycoplasma are pathogenic in animal and plants. They used to cause diseases in plants and animals. Okay, so this is the structure of mycoplasma, smallest living organisms known. Let us continue with the topic, kingdom Protista. So far we discussed about the kingdom Monera, where we discussed about the body organization and cell structure and types of bacteria. All these things we studied. Similarly, in kingdom Protista, we are going to discuss about its habitat and the type of cell and different classification which is observed in the kingdom Protista. Okay. Now, when it comes to the habitat where they live, most of them are aquatic forms. Okay, Protestant members are aquatic, most of them. Some of them are parasites, some of them are saprophytes. So there are different types, but mostly they are aquatic. Now, we should have a general idea. What are these Protestant members? All the unicellular eukaryotic organisms are considered as Protista. Now, suppose there is one organism having exact same character of a plant and one organism having exact same character of an animal. Another organism is having exact character of a fungus. But if it is unicellular, then they are eukaryotes, then they are placed under Protista. That is the criteria for the classification. So here we observe diverse organisms. Okay. Now, boundaries of this kingdom are not well defined. They are single-celled eukaryotes, but their boundaries are not well defined. The reason is the same thing. Just because they are unicellular eukaryotes, even though they have a similar character like plants or animals or a fungus, now they are classified under kingdom Protista. Now, members of the Protista are primarily aquatic, and being eukaryotes, Protestant cell body contains well-defined nucleus. Of course, they are eukaryotes, so they have a true nucleus, and apart from this, they have all the membrane-bound cell organelles like mitochondria. Some of them they have chloroplast, and they have true nucleus, endoplasmic reticulum, Golgi complex. All those things are observed in the cell. Now, protists reproduce asexually and sexually by process involving cell fusion and zygote formation. So that is the mode of reproduction observed in them. Okay. Just observe this slide, a beautiful slide showing diatoms. So this is how diatoms, if you take a sample of water and observe it under microscope, it appears like this. Okay. These are desmids. This is the structure of one desmid. Now, coming to the classification of Protista, first we will study chrysophytes. Okay. So group includes diatoms and golden algae. Those are the desmids. Here, let us go by the character one by one. They are freshwater as well as marine water. They are found in freshwater as well as marine water. And they are planktons, passively float on the surface of water. So they are called planktons. There are so many classifications like planktons, benthos, nectos. So depending on their location in the water. So these organisms, they freely float on the surface. So they are considered as planktons. Now, most of them are photosynthetic, means they are autotrophs. They have an ability to synthesize their own food. Diatoms, the cell wall form two thin overlapping shells which fit together as in the soap box. That means here cell wall is having two plates. One fits firmly with the another, and cell wall is made up of indestructible material, that is silica. Here there is one important character. When these diatoms they die, their indestructible cell wall, it remains as it is. For billions of years, they get deposit in the bed of the sea. So they form diatomaceous earth. This is one of the important characteristics of chrysophytes. Okay, their silica cell wall is indestructible and get deposited at the bottom of the sea, forming a diatomaceous earth. Now, what is the importance of this diatomaceous earth? This can have some economic importance. That diatoms, that silica, what is left behind after their death, can be used to filter the syrups and oils. They are slightly gritty in nature, can also be used as a polish. Okay, so that is the economic importance of diatomaceous earth. Now coming to the next one, just observe this slide. Both the slides are very beautiful. One slide, here you can see the red tides, and another is showing bioluminescence. This is Goniolix, massive growth, and this is Noctiluca. So these two organisms, they belong to dinoflagellates. Just observe the name of the classification, dinoflagellates, means they have two flagella. Just observe their location. Before that, they are mostly marine and they have different colors, yellow, green, brown, blue, red, depending on the main pigment present in their cells. And cell wall has stiff cellulose plates on the outer surface, and this two flagella what they have, one lies longitudinally and other is transverse. Why they have two flagella means, one longitudinal flagella, suppose this is the organism, now one flagella is present here, so this will help in the forward movement, and another flagella is present here, that is transverse, it helps in rotation. Okay, so this is how they have two flagella and they use those flagella during locomotion. And very often these red dinoflagellates, they undergo massive reproduction and produces millions of organisms, and they form a red tide. Okay, here in this picture I have shown, this is how they form a red tide. And when they grow massively in the ocean, to their surrounding, it releases a toxic chemicals, and that is lethal to aquatic organisms. So the fishes nearby, the overgrowth of these dinoflagellates, they die. Okay, and this is about the dinoflagellates. Next one, euglenoids. Just observe the picture. One is diagrammatic representation, another one is the slide show. Euglena, a very common example we studied in our lower class also. When we look at this organism, it almost looks like a plant related organism, right? It looks like a plant. Yes, of course it is green, autotrophic, it has a pigment similar to higher plants, and it can show a photosynthesis. So this Euglena looks like a plant, but it is unicellular, so they are classified under euglenoids, which comes under the Protista. Now just look at the organism here. It has one long flagella, another one small flagella is present. This is the mouth, we call it as gullet. Now it has true nucleus and all those things, chloroplast. Now very important, the cell wall is absent here. Instead of that, they have a proteinaceous covering called pellicle. Okay, so they are aquatic. Majority of them are freshwater forms, and instead of cell wall, they have a protein rich layer called pellicle, and which makes their body flexible. Okay, and they have two flagella, one short and one long. See, this is very important character, that one long flagella helps in locomotion, and short flagella, which is present near the gullet, helps in capturing the prey. So when it comes to euglenoids, we think that it is completely autotrophic, but the fact is different. Euglenoids, whenever there is a sufficient sunlight, they uses the sunlight and undertake the photosynthesis. But when sunlight deprived, they show heterotrophic mode of nutrition. That means they prey on the smaller organisms. That means here they have photosynthetic as well as heterotrophic mode of reproduction, mode of nutrition. So it is called mixotrophic mode of nutrition. Very important character. Euglena shows mixotrophic mode of nutrition. Now, the pigments of euglenoids are identical to the pigment which is present in the higher plants. Okay, that is one more character. Now, just look at this picture. This is the fallen wood, or you can say tree trunk, and above this you can see the massive growth of slime molds. This is the slime mold, a beautiful picture of a growth of the slime mold. When we look at this slime mold, it almost appears like a fungus, and most of the nature is also like a fungus. But there is a fundamental difference. Fungus has chitin in its cell wall, but this organism doesn't have cell wall. That is the main difference. Now coming to character, these are saprophytic organisms. They grow on the dead and decaying material. Okay, body moves along the decaying twigs and leaves, engulfing the organic material. So they take the organic material by engulfing. Okay, that means they degrade it. So under suitable conditions, they form aggregations called plasmodium. So just now you saw the picture, the massive growth of plasmodium. This is under the favorable condition. This plasmodium produces the fruiting bodies. So they grow above the surface of the body of this slime molds. This fruiting bodies produces spores, and these spores are meant to survive in the unfavorable condition. So very important character here, spores have true cell wall. The body of the organism doesn't have cell wall, but spores have true cell wall so that they can survive in the unfavorable conditions. Okay, on set of favorable conditions, they germinate and give rise to a new body. Now, how they spread to new areas? Through the wind. Okay. The next one is protozoa. Now, again in protozoa, there are different organisms. We'll study in detail. But protozoan members are heterotrophs. Okay. Now, here some of them are pathogens and some of them are parasites. Let us take the first example, that is amoeboid protozoans. The best example for amoeboid protozoa is Entamoeba histolytica. So what they cause actually, where they are present? This Entamoeba histolytica commonly live in fresh water, sea water, or in moist soil. Okay. If the food is contaminated with the Entamoeba histolytica through any of this medium, then if we consume them, it causes amoebic dysentery in our body. Okay. So it is a parasite. And one more thing, remember the marine forms of this Entamoeba histolytica has silica on its body. Now coming to next one, flagellated protozoans. Best example for flagellated protozoan is trypanosoma. It used to cause disease, sleeping sickness. Okay, so the members of this group are either free living or parasites, and they have flagella, so the name flagellated protozoans. The parasitic forms causes disease such as sleeping sickness. Okay, example trypanosoma. Next, ciliated protozoans. Example is paramecium. Let me show you the picture of the paramecium for better understanding. This one is the trypanosoma, which causes sleeping sickness. Okay, so this is paramecium. Just look at the body of the paramecium. Here you can see cilia, and this is the oral groove, or we can also call it as gullet. Okay, there are two type of nucleus, micronucleus and macronucleus, very important character. Okay, so one helps in the functioning, normal functioning of the body of the organism. Another helps during the division of the cell, that means during the reproduction. Now, when this cilia move, it helps in the movement of the organism. At the same time, it collects the water, or it pushes the water towards this oral cavity or gullet, so that it can trap the smaller organisms and engulf it. Okay, this is how paramecium function. Now this one is another one, we call it as sporozoans. So sporozoans, best example is Plasmodium vivax. So it is a parasite which used to cause malaria in human beings. Okay, so these are some of the examples for kingdom Protista. So what and all we studied? Chrysophytes, example is diatoms and desmids. Then dinoflagellates, best example is goniolix, which forms redditide. And then euglenoids, euglena, and which shows mixotropic mode of nutrition. And slime molds, they produce plasmodium on favorable condition. On set of unfavorable condition, they produce the spores, which has true cell wall. And then we studied protozoans, which includes amoeboid protozoans, ciliated protozoans, flagellated protozoans, and sporozoans. So this last part mainly they ask in match the following type of questions. In rest of the classifications, in your competitive exam, they give all the characters. Say for example, characters are given like this. Organism is found in fresh water. They show a pigment which is similar to higher plants. Cell wall is absent. Instead, they have proteinaceous layer that is called pellicle, and they have two flagella, one is large and another is short, and they show a mixotropic mode of nutrition. This type they have given the character. On this character, they will ask which this character matches which of the following organism. You can say it is euglenoid if we know the characters properly. Okay, this is how questions will be framed. Or they will ask, select the correct statement or incorrect statement. Say for example, all the characters are given for dinoflagellates. All the dinoflagellates characters are given, but they slightly change one character. There they have given one flagella is longitudinal, one is transverse given. So here what they will give, they have two flagella, both are present in the longitudinal. Then that statement is wrong. Okay, so they will slightly change the words while forming such type of questions. The topic is kingdom fungi. See, kingdom fungi, it's very common. Wherever we go, we observe certain type of fungus. In rainy season, it's very common. They usually grow in the warm and humid places. Okay, so if you place any of the food like citrus fruit or banana or any type of bread outside for some interval of time, and you observe certain dust-like substances growing on it, it is actually a fungus. Okay, fungus, they all of them are heterotrophs. They cannot synthesize their food. Okay, so they have to depend on dead and decaying material, or sometimes they can act as a parasite. Sometimes they show a association with algae, and that we call it as lichens. We study the character of fungus one by one in detail. Okay, let us start with its habitat where it grow. Okay, the fungi constitute a unique kingdom of heterotrophic organisms, means they don't have ability to synthesize their own food, and they show great diversity in morphology and habitat. Why they have taken morphology? See here, we can see very small fungus like yeast, which is unicellular, and others are filamentous forms. Like some of them are forming a hyphae, and hyphae form a network like structure called mycelium, and these mycelium produces fruiting body, and that fruiting body has different structures, sac like structure, basidiocarp like structure. So there are different types of fruiting bodies are produced. Okay. And habitat, as I said, some of them grow on the rotting fruits, rotting food, and some of them grow on the body of the living organism as a pathogens, and some of them they cause disease in plants like wheat rust. These are the disease caused by fungus. Okay, and you must have seen fungi on moist bread and rotten fruits. And the common mushroom you eat and toadstools are also fungi. Here, common mushroom we all seen. It is a white colored button mushroom. Have you seen toadstools? Actually, they are reddish and very attractive, but they are poisonous. Okay. The white spots seen on the mustard leaves are due to the parasitic fungus. So mustard leaves get diseased by parasitic fungus. So some unicellular fungi like yeasts are used to make the bread and beer. So why they have given all these sentence? Just to have a general idea about the fungus where we commonly observe them. Okay. And the fungi cause disease in plants and animals. Wheat rust causing Puccinia is an important example, because we know that most of the part of our country where they grow wheat, and there the most disturbing disease is wheat rust by Puccinia. Okay. Then some are the source of antibiotics. You know that the organism Penicillium notatum is used in the extraction of the antibiotic penicillin. It is an accidental discovery. Scientists, when working on the culture medium of bacteria, accidentally they kept the culture medium without wash. So next day when they come back, they observed that the growth of bacterium got restricted due to the growth of fungus. So when they get extracted the component from it and they tested it on the growth of the bacterium, they got to know that the Penicillium notatum fungus produces the antibiotics which inhibits the growth of bacteria. That is how accidentally they found the antibiotic. Okay, and fungi are cosmopolitan. You found fungus in different places like in air, water, and in soil, and on the animals and plants. So they prefer to grow in warm and humid places. The best condition for the growth of fungus is warm and humid places. Now, with the exception of yeast, which are unicellular, yeasts are unicellular, remember this, fungi are filamentous. So they produce slender thread-like structures called hyphae. Here there are so many characters you need to remember. Hyphae is the body of the fungus. Okay, this hyphae sometimes without branch, sometimes they are branched. And hyphae, if they show the cross walls, then it is called septate. If cross walls are absent, then it is called aseptate. Some members of the fungus, they show aseptate hyphae where many nucleus is present, and that condition we call it as cenocytic condition, aseptate cenocytic condition. Okay, so here the same thing is mentioned. Network of the hyphae is known as mycelium. Very important character. So if you have observed anywhere how the mushrooms are cultivated, they take a small hyphae and they place it in the medium, nothing but the nutrient medium or the dead and decaying material. On that, this hyphae, they grow massively and produces white thread-like structures. That is called mycelium. Okay. Now, the hyphae are continuous tubes filled with multinucleated cytoplasm, and these are called cenocytic hyphae. Very important distinguishing character observed in one of the classification of fungus, that is Phycomycetes. Now, others are septate, means having cross wall in their hyphae. The cell wall of the fungi are composed of chitin and polysaccharides. That makes the fungus different from other organisms. One of the criteria used for classification of fungus, that is cell wall composition. It is made up of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from the dead substrates and hence are called saprophytes. Here also we observe diverse mode of nutrition. One is saprophytic mode, and those that depend on the living plants and animals are called parasites. Nothing but some of them they do not cause disease, some of them they cause the disease. Okay. Then they can also live as a symbionts in association with algae, then it is called lichens, and with the roots of higher plants like pinus, it is called mycorrhizae. See, in the case of pinus grow in the area, mainly hill stations, where the roots of the pinus plant cannot reach to the depth to absorb the water, or the retention of water in the soil of the hill station is very less. At that time, they make association with the fungus like mycorrhizae. They grow deep into the soil and absorb water and return it to the plants. At the same time, plants provide food to the organisms. So mutually these organisms are benefited. So they are the symbionts. Now, reproduction in fungi can take place by vegetative means, by fragmentation or fission and budding. So all the three types are observed here. Asexual reproduction is by spore and conidia. See, all these spores are very, very important for competitive exam. Please do remember. Asexual spores called conidia, exogenously produced, or sporangiospores or zoospores. Okay, sporangium produces sporangiospores. They are endogenously produced inside a sporangium. Later I will show the figure. Okay, zoospores are motile. And sexual reproduction by, again you should make clear difference between the asexual spores and sexual reproduction by the other spores like oospores, ascospores and basidiospores. There is a reason to call these spores with these names. Ascospores are produced inside the ascocarp, sac-like structure. Basidiospores are produced inside the basidium, so the name basidiospores. So the various spores are produced in distinct structure called fruiting bodies. It's very common. If we consider this is the hyphae, septate hyphae. Okay. Now this septate hyphae produces fruiting body like this. And here you can see the fruiting bodies. This is how we can see the fruiting bodies. And that this is actually a structure of a agaricus which produces basidiocarp, inside which basidiospores are present. The sexual cycle involves the following three steps, very important. In sexual mode of reproduction, there are three steps are present. One is fusion of cytoplasm, that is called plasmogamy. Once the cytoplasm fuses, two nucleus from the two cells, they fuse immediately, and that process we call it as karyogamy, fusion of nucleus. Zygote formed are diploid, but there is no diploid life cycle in fungus. So the zygote immediately undergo meiosis and produces 4 haploid spores, and they are haploid, they give rise to haploid body of the fungus. Now when fungus reproduce sexually, two haploid hyphae of compatible mating types come together and it fuses. In some fungi, fusion of the two haploid cells immediately results in the diploid cells. Okay, this is very common in most of the fungus. But in some of the members of Ascomycetes and Basidiomycetes, it is very common in Basidiomycetes. First two compatible hyphae, they come near to each other, and cytoplasm of these two hyphae fuses, leading to plasmogamy. And now this hyphae, when it is fused, two nucleus are present, but these two nucleus immediately will not undergo fusion, so it will be delayed. In that situation, in one cell we observe two nucleus, and that is called dikaryon phase. Very evident and important character of Ascomycetes and Basidiomycetes. So this dikaryon phase is not permanent. After some time, again nucleus fuses, and that process we call it as karyogamy. Karyogamy is followed by zygotic meiosis. Okay. Then the later parental nuclei fuses and the cell become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to the formation of haploid spores. Very important, even though sexual reproduction lead to the diploid cell zygote, after that zygote undergo meiosis and the spores produced in fruiting bodies are haploid. Okay. So the morphology of the mycelium, mode of spore formation, this is very, very important character. Morphology of the mycelium, mode of spore formation, and fruiting bodies form the basis for the kingdom into various classes. So based on this character, further fungus are classified into Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. We will study one classification in detail. So the first one is Phycomycetes. So usually in classification, first we study most primitive and later we start to study the advanced one. Now Phycomycetes is the most primitive form of fungus. Here most of them are found in aquatic habitat or on the moist wood, or sometimes they found in the found as an obligate parasites on the plants. The important character of the classification Phycomycetes is mycelium is aseptate and cenocetic. That is the most distinguishing character. Again, all these characters are very, very important in the preparation of NEET exams, because there mostly they ask statement based questions. Okay, so you should be very thorough with the important characters of classification. One is aseptate and cenocetic. Asexual reproduction takes place by zoospores, they are motile, and aplanospores, they are non-motile. And these spores are endogenously produced in the sporangium. Let me draw one small diagram to understand this. This is the fruiting body. Here it produces the sporangium, inside which spores are produced. So this is nothing but endogenously produced spores. Endogenously produced spore. Okay. Now, zygospore is formed by the fusion of two gametes. This is very, very important. By the sexual mode of reproduction, it produces the zygospore. So these gametes are similar in morphology, then it is called isogametes. Dissimilar, either anisogamous or oogamous type. All these things are basics. Isogamous means gametes are of similar sized. Anisogamous means one large, one small. Oogamous means one is static, large, another one is small and motile. So this type of gamete formation is observed in sexual mode of reproduction of Phycomycetes. Some common examples are Mucor, Rhizopus and Albugo. You can remember it as RAM. Rhizopus, Albugo and Mucor. Okay, all these examples are important. Here Rhizopus is the common bread mold and Albugo, it's a parasitic fungus on mustard. Very, very important character. Parasitic fungi on mustard. This can be asked in match the following type of questions. Now this is the diagram. One is Rhizopus, another one is Mucor. These are the example. Here you can see the sporangium inside which spores are produced. This is the hyphae and this is the fruiting body. Upper part is the fruiting body. Here you can see the mycelium on the bread. Okay. Now coming to next classification, that is Ascomycetes. Listen, this is again a very important classification. Commonly known as sac fungi. They are commonly known as sac fungi, mostly multicellular. Example penicillium, rarely unicellular. Example is yeast. So common baker yeast belong to the classification Ascomycetes. They are saprophytic, means growing on dead and decaying material. Decomposers, means helps in recycling of minerals. Parasitic, grow on other organisms. Coprophilous, grow on the dungs. And mycelium is branched and septate, very important character. In Phycomycetes it is aseptate and cenocytic condition, but here it is branched and septate. The sexual spores are conidia. Now what is the difference between sporangiospore and conidia? Conidia are produced exogenously, sporangiospores are produced endogenously. So here conidiophore is the fruiting body on which conidia are produced, so they are exogenously produced. I will show that picture. Okay, exogenously produced on the special mycelium called conidiophores. Conidia on germination produces the mycelium. Sexual spores are called ascospores, which are produced endogenously in the sac-like asci. Again, in statement based question they may twist the statement like this: ascospores are the asexual spores produced endogenously. No, it is wrong, because ascospores are the sexual spores produced endogenously in asci or cup-like structure. Okay, remember, very important statement. And these asci are arranged in different types of fruiting bodies called ascocarps. Some examples are Aspergillus, Claviceps, Neurospora. See, whenever they have given example, it should have some economic importance. We know yeast is used in preparation of bakery items and some alcohol preparations like beer. And penicillium, it is a fungus from which antibiotic is extracted. Then Neurospora, it is extensively used in genetic studies. See, in scientific studies, we use Drosophila melanogaster and guinea pigs, rats, then pea plant. Similarly, in fungus, they take Neurospora for the extensive genetic studies. And we also have example like Aspergillus. which used to cause some of the respiratory diseases. Okay, it's commonly observed on onion. Then many members like morels and truffles are edible delicacies. Here actually morels and truffles are very costly fungus, which cost thousands of dollars. Okay, let us see all the pictures of the fungus to get detail about this. See the beautiful picture of this slide. Whenever I look at these amazing pictures, I wonder how these organisms are. This is yeast, unicellular. Penicillium notatum. Here you can see the conidia formation. This is the conidiophore. Conidia are exogenously produced. This is Aspergillus niger. Here also you can see the conidia produced exogenously. And this is I think Neurospora. Sorry, this is Neurospora. This is Claviceps, and this one is Morels. Okay, this is Morel, and this is Truffles. Commonly found in pinus forest. They are very peculiar fungus, not found commonly in all the places. They are very unique and found in the pinus forest. Okay, so their value is very, very high. Very costly fungus it is. Okay, not easily, you cannot cultivate it. Next one, Basidiomycetes. Very common fungus. Even we ate it. Edible agaricus is the best example to get imagination of all the characters. Now, Basidiomycetes is commonly known forms of Basidiomycetes are mushrooms, bracket fungi, and puffballs. Bracket fungi is very common whenever you visit any of the forest or maybe nearby your house. On the tree trunk we observe certain very hard or woody like mushrooms. Those are the bracket fungi. And puffballs, not very common. In some forest area you can observe this. It looks like a ball. It is having millions of spores. When it completely matures, if you hit that fungus with any of the hard material, these spores get liberated like anything. Okay, that is the puffball. So they grow in soil, on the logs and tree stumps, and live in living plant bodies as parasites. So rusts and smuts. The mycelium is branched and septate. So there is a reason to name it as rust and smut. Okay, rusts are actually obligate parasites. Puccinia is the best example. Ustilago is an example for smut. It is a facultative parasite. If it has the host, then it grows along with the host. If not, also it can survive. So it is called facultative. If the relationship is compulsory, then it is called obligatory relationship. Okay. Then asexual spores are generally not found. Very, very important character, distinguishing character. Asexual spores are found in Phycomycetes, that is the sporangiospore. Asexual spores are found in Ascomycetes, that is conidia. But in Basidiomycetes, asexual spores are absent. Then the type of spores observed in Basidiomycetes, all are sexual spores. So where they are produced? They are produced in the basidiocarp. How they are produced? By the sexual mode of reproduction. How the sexual mode of reproduction takes place? This is very, very interesting. When fruiting bodies are produced, two compatible hyphae, they come close to each other. Plasmogamy takes place. But nucleus, they immediately not undergo fusion. So dikaryon intermediate phase is formed, and then followed by fission of nucleus. that is karyogamy. Once the diploid zygote is formed, diploid zygote undergo meiosis and produce 4 haploid spores, and those haploid spores on germination give rise to hyphae. So these spores, basidiospores, formed by sexual mode of reproduction, are exogenously produced on the basidiocarp. These are the most important characters of basidiocarps. Now, some of the examples are Agaricus, Ustilago and Puccinia. So this is the bracket fungi. This is the wooden log on which bracket fungi is growing. This is puffballs. Agaricus, very common one. This is the Puccinia. Last one, but this is very interesting classification, Deuteromycetes. When we read in NCERT, it seems to be very confusing. Why they have given like this? They are called imperfect fungi. Why they are imperfect? All those things we will discuss now. See, Deuteromycetes, in this classification, the fungus included are, they don't have sexual mode of reproduction, or so far their sexual reproduction are not identified. So in this case, what happens, whenever in the process of research, if they find out any of the members of Deuteromycetes, they have sexual mode of reproduction, then immediately those organisms are taken out from the Deuteromycetes, either placed in Ascomycetes or in Basidiomycetes. Okay, this is very important character. So here there is no sexual mode of reproduction. Then how they reproduce? They reproduce mainly by asexual mode of reproduction, that too by production of conidia. So conidia is the asexual spore here, and they are the main decomposers. Most of them are decomposers. Okay, so here examples are Alternaria and Colatotrichum and Trichoderma. Let me show you some of the pictures. This is Trichoderma, and this is Alternaria, and this is Trichoderma viridae. Okay, nowadays most commonly used in preparation of fertilizers. Okay, now so far we studied 3 kingdoms: Kingdom Monera, Kingdom Protista and Kingdom Mycota. The last part of the topic, which is virus, viroids, prions and lichens, these 4 classifications are not belonging to any type of the kingdoms. Okay, we will study this in detail but separately. Now, virus are non-cellular organisms. To define it as a cell, it should have some characters, but virus did not fulfill any of the characters of the cellular body. So they are non-cellulosic or acellular, you can say. Now we know that virus causes different types of disease. All of them are obligate parasites. They are pathogens. They used to cause disease. Okay, here the body of the organism is not much complex. It is made up of genetic material, DNA or RNA and protein. That's it, nothing else. Okay, so virus are acellular and made up of protein and genetic material. This genetic material may be DNA or RNA. So many scientists, they started working on the on depicting the structure of virus. It all got started in 1892, long back. Okay, here Dmitry Ivanovsky in 1892 first coined the term virus, means venomous, poisonous fluid. How he recognized? Actually, when he was working on tobacco plant, he found out the organism which is causing tobacco mosaic disease is tobacco mosaic virus, and they passes through the bacterial filter paper. Okay, so that means they are not bacteria, they are something else, and they are the virus. In 1898, after 6 years of the contribution of Dmitry Ivanovsky, M.W. Bezernik, what he did, he took the sap from the infected plant and he injected it to the healthy plant. Now healthy plant got the disease. So he named it as Contagium vivum fluidum. It is a poisonous fluid, okay, infectious living fluid. And W.M. Stanley in 1935 showed that viruses could be crystallized, and crystals consist largely of proteins. Okay, they are inert outside the specific host, and viruses are obligate parasite. That means outside the host they are inert. They cannot show any metabolic activity. They are acellular. Only inside the host they become parasitic. Okay. So in addition to protein, viruses also contain genetic material that could be RNA or DNA. No virus contains both DNA and RNA. This is a very important character. Now, the virus is a nucleoprotein and the genetic material is infectious. They are nucleoproteins. That means they have nucleic acid plus protein. Remember, they are not same. The virus which causes disease in plants, they have genetic material that is single-stranded RNA. And the virus which causes disease in bacteria, they are called bacteriophages. They have double-stranded DNA. And virus, they cause disease in animals. They have single-stranded RNA, double-stranded RNA, or double-stranded DNA as their genetic material. So this is the very, very important character, distinguishing character of the virus which causes disease in plant, animal, and bacteria. Okay. Mostly they are asked in match the following type of questions. Now the protein coat called capsid, made of small subunits called capsomeres, and protects the nucleic acid. These capsomeres are arranged in a helical or polyhedral genetic form. So if you observe the tobacco mosaic virus, TMV virus, and a bacteriophage, in TMV virus, protein coats are in the form of capsids. In bacteriophage, it is in the polyhedral geometric forms. Okay. Now viruses cause diseases like mumps, smallpox, herpes, and influenza. These are the common disease caused by the virus. AIDS in humans is also caused by virus, acquired immunodeficiency syndrome. In plants, symptoms can be mosaic formation, leaf rolling and curling, yellowing, vein clearing, and dwarfing, stunted growth. See, all these are the symptoms. Here, mosaic formation means there is no proper structure to the leaf. It is a distorted leaf. Then leaf rolling, in the sense it curve inward back. Okay. Then and curling, same thing. Yellowing, leaf become yellow. And vein clearing, that means the inner part of the veins are completely affected by virus and it is transparent almost. And dwarfing, small, stunted growth. Okay. These are the symptoms observed by the disease caused by virus in plants. Now coming to the next classification, that is viroids. Very interesting organisms. In 1971, T.O. Danier first identified this in potato spindle tuber disease. Surprisingly, this organism has only genetic material, nothing else. That is RNA. And this RNA has very low molecular weight, very important character. Okay. And they are smaller than virus. Next one, prions. Again, they are surprising organisms. See here, the disease causing organism has some foldings of protein. That's it, nothing else. This folding of protein causes disease. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded proteins, nothing else. And the agent was similar in size to viruses, and these agents were called prions. They used to cause disease in cow and human beings. In cow, it is called mad cow disease. The scientific name is bovine spongiform encephalopathy, in short from BSE. And human beings, it causes Creutzfeldt-Jakob disease, and it is called CJD. Okay, these are the disease caused by prions. The last part of the topic, lichens, my favorite part, because lichens are beautiful organisms. They grow in the area where pollution is very, very less. They are the best pollution indicators. If there is a pollution, these organisms disappear from the surface of the plants. So these lichens are the association of two organisms. One is fungus, another one is algae. Okay, green algae and fungus, they show the association and form a lichens. Fungal component is called mycobiont, algal component is called phycobiont. Here we know that fungus are heterotrophs, algae are autotrophs. So here fungus gives the place to algae, in turn algae provides food to the fungus. It is a symbiotic association. Okay, they commonly grow on the bark of the tree and found in area where pollution is very less. If pollution occurs, they disappear from the surface. They are the very good pollution indicators. And usually these lichens, they appear on the bare rocks, and they play very important role in the succession. Okay, so this is the picture of lichen growing on the branch of the tree. Okay, yes, with this we completed the topic biological classification.